الأنبياء والمرسلين نبينا محمد وعلى آله وصحبه أجمعين. Assalamu alaikum everyone. Today I'll be giving you a review session on the assimilation of nutrients lecture from the physiology part of the GIT. This lecture basically tackles how different nutrients get digested and absorbed. So we're mainly going to be talking about carbohydrates, proteins, lipids, and then some like assorted stuff, just real quick. All right, so bismillah. So the start of the lecture, just basically we need to lay some ground rules, which is the fact that in the small intestines, mainly the small intestines is where the absorption of nutrients happens, right? So in the small intestines, we have the villi and we have the crypts. The stem cells that make all the epithelial cells that will become absorb, that will start to absorb and will start to secrete and absorb, the stem cells are all present in the crypt, but not at the bottom of the crypt. No, it's just near the bottom of the crypt you will find the stem cells. These stem cells like replicate very, very, very rapidly, and like the entirety of the epithelium here will basically like replace itself every like 3 to 6 days almost. And the blood supply is usually better here than here. So like the closer you get over there, the less the blood supply, and then the cells will die, and then they will be exfoliated into the into the lumen and degraded in the lumen. Okay. You'll find here, the stem cells. You'll find the Paneth cells over here, which I believe are more have a bigger immune system significance. And like these are epithelial cells that will be absorbing stuff. The interendocrine cells that will be secreting different hormones, and you have the goblet cells as well. All of them are coming from the stem cells over here, and they just go up like a conveyor belt, basically. Okay, so basically the upper part of the villus is the most functional part of the villus. Everything below will be like less functional, less functional, less functional. Is there any questions about this part? I'll basically be asking after every slide or every small topic if there's any questions, and then I'll go forward like if like nobody asks in like 10 seconds. Okay, so that's how I usually deal with it. Right, bismillah. So we will start by talking about the different nutrients, and we're starting with carbohydrates. Carbohydrates get basically digested and absorbed in the they get digested and absorbed, and the digestion of carbohydrates happens both in the lumen and on the brush border of the microvilli. Okay, the cytoplasm does not have any enzymes that will digest or like will break down the carbohydrates. Okay, and the main thing we're looking at when we're talking about the luminal digestion, is the pancreatic alpha amylase. And when we're talking about the microvillus digestion is the saccharidases on the brush border enzyme. And we'll talk about them further right now. So carbohydrates, the important thing to note about them is that they can only be absorbed when they are monosaccharides. So just a pure glucose, a pure fructose, galactose, so just one monosaccharide. That means we'll need to break down everything until it becomes a monosaccharide. Okay. Now, most of the glucose or most of the carbohydrates that we will find in food will come in the form of like polysaccharides. Mainly the ones that that are useful to us are starches. So starches that come from like rice, potatoes, etc., will be basically polysaccharides of glucose in a line like this. All right, and these are easily digested and absorbed. The main ones that we need to know are amylose and amylopectin. Amylose is just one line, just no branching, no nothing. It's just 1,4 glycosidic linkage, as you can see here. So between the carbon number 1 and carbon number 4, carbon number 1, carbon number 4, as you can see here. Okay, and it's just one line. The other one is the branched polymer, which is the amylopectin. The amylopectin goes both 1,4 and it branches at 1,6. Okay. Now, just to note, when you're looking at this, the one between these two that is broken down or like gives you more energy immediately and very fast is of course the amylopectin one, because the enzymes will not only be working at the main strand, but they will be also working at every single branch. So like every single branch will give you glucose immediately, while here you have to work like one by one by one by one. So it takes much longer to get energy from amylose than it takes from amylopectin. Okay, and this is why basically our glycogen in the body is more similar to amylopectin, but it's even more branched than amylopectin, which means that you can get energy from it even faster. Okay, so that's when we talk about starch. So starch are polysaccharides that we can digest and we can absorb, and we will talk more about how we absorb them, and they are amylose and amylopectin. On the other hand, we have cellulose. Cellulose is another glucose polymer, just like amylose is a glucose polymer. Cellulose is as well, but it, rather than having an alpha 1,4 glycosidic linkage, it has a beta 1,4 glycosidic linkage. And so our bodies are not equipped with enzymes that are capable of breaking down this beta 1,4 glycosidic linkage. And so we cannot get the glucose from cellulose. So we cannot eat grass, basically. That's what we're saying here. Okay, these are the fibers that we know. Now let's talk more about like the details of how we digest. Okay. Do we have any questions until now about the amylose and amylopectin and cellulose? All right, next. All right, so amylose and amylopectin and how we break them down. Amylose, again, it's just simple 1,4 linkages. Okay, one alpha 1,4, alpha 1,4. Amylopectin has alpha 1,4, as you can see here, and has this alpha 1,6. Okay, so for the alpha 1,4, we just need the enzyme alpha amylase. Alpha amylase can break down any alpha 1,4. Okay, but one thing about alpha amylase is that it is an endoenzyme. What do we mean by an endoenzyme? It means that it can only break down a linkage in the middle of the polymer. It cannot break down, or it cannot degrade like the edges of it. So it cannot break down here. Okay, and like at the end over there, it cannot break down over there. It can only break down stuff in the middle. All right, it cannot break down the terminal ones, only internal ones. Okay, so that means that the alpha amylase will always leave the amylose in the form of maltose, which is glucose plus glucose, because now this is the terminal one. The alpha amylase cannot break this, and maltotriose, which is 3-glucose, and again it cannot break them because this one is a terminal, and this one is also a terminal. Okay, so this is when we're talking about amylose. This is the breakdown of amylose. On the other hand, amylopectin, we will need, when we break it down, basically we break down with alpha amylase this area and this area. Okay, and this will leave us with the area right here, which has the 1,6 linkage. So any polymer of glucose, all right, that has both 1,4 linkage, as you can see here, 1,4 linkage, 1,4 linkage, and 1,6 linkage at the same time, we call it an alpha limit dextrin. Okay, so this is a limit dextrin that goes 1,4, 1,4, 1,4, 1,6. Okay, and this is our maltotriose and maltose. Okay, so again, just by the work of alpha amylase alone, amylose will be turned into maltose and maltotriose. Amylopectin will be turned into maltotriose, maltose, and alpha limit dextrins. Is this clear? Okay. So alpha amylase is an endoenzyme, and it does not produce glucose. We cannot get glucose simply from alpha amylase. Amylopectin is not clear. So amylopectin, again, because it is alpha 1,4 over here, alpha 1,6 over here. Okay, so our alpha amylase can only break alpha 1,4 linkages that are not terminal, so they are in the middle. So the alpha amylase will come and break down over here. Okay, it can also break down over here, but it can break down over here or here. Okay, so once it broke down this, and then it broke down this from this side, you will be left with this shape, this Tetris shape thingy. Okay, this shape that has alpha 1,4 over here, that's 1,4, 1,4, right? These are 1,4 linkages, and this is a 1,6 linkage. this shape right here, because it has both alpha 1,4 and alpha 1,6, we call it a limit dextrin. Okay? The rest of it, everything else, basically everything else, because this is just 1,4s and this is all 1,4s, so they will be turned just like an amylose to be turned into a maltotriose and a maltose. The main point that you need to know is that the degradation of amylopectin by the amylase enzyme will result in limit dextrins. Okay, this is the main thing that differentiates between amylose and amylopectin. All right, and again, glucose is not a product, only maltose, maltotriose, and limit dextrins. Right, now what do we do with these maltotriose, limit dextrins, maltose, whatever? Okay, this is what we're talking about right now. We have multiple enzymes that can work this work. So we have this maltase glucoamylase and this sucrase isomaltase. Okay, so looking at the maltase glucoamylase, this one right here, this one simply just takes the maltose and maltotriose, these ones, and just breaks them down into glucose. So the maltase glucoamylase, this is an exo. This is an endoenzyme. This is an exoenzyme because it can cut the terminal bonds. Make sense? Simple enough, I think. So it cuts the alpha 1,4 terminal bonds of maltose and maltotriose. Simple. That's what maltase glucoamylase does. And this is where are we talking right now? We're not in the pancreas or not the pancreatic secretions. This is the brush border. Okay. On the other hand, alpha limit dextrins have alpha 1,6. All right. So how do we break down this alpha 1,6? We can only break down the alpha 1,6 via the enzyme isomaltase right here. Isomaltase will take this alpha limit dextrin and break down the alpha 1,6 in it. So basically, look at this. It breaks down this over here. So you are left with 2, that's a maltose, and a 3, that's a maltotriose right here. Maltose, maltotriose. Okay? So this is basically the full breakage or the full degradation of amylopectin. So the full degradation of amylopectin is first, alpha amylase will leave it to limit dextrin, maltose, and maltotriose. Then the limit dextrin will be broken down by the isomaltase in the sucrase isomaltase complex, and it will be turned into maltose and maltotriose. And then the maltose and maltotriose will be turned into glucose via the maltase glucoamylase. Is this clear? Okay. And again, this is happening in the brush border. The amylase step is in the pancreas. Next is other stuff other than glucose. We have sucrose, we have lactose. These are disaccharides that are not just glucose mixed together. So for sucrose, we have this sucrase isomaltase. It's a complex of 2 enzymes stuck together. The isomaltase works on this step. The sucrase works on sucrose, breaks it down into glucose and fructose. Simple enough. And lastly, we have the lactase, which breaks down lactose into glucose and galactose. All of these are on the brush border. So any disease in the enterocytes themselves will result in problems of carbohydrate digestion, and you will have a lot of disaccharides and alpha limit dextrins. Okay? Any problem in pancreatic secretions will have problems in this first step. So we will have like a full amylopectin that is not degenerated at all. Okay? So this is it for this part. Now, one thing you should like have in mind is that carbohydrate, like the secretions of all these enzymes is like very, very high. So like the pancreas releases so much amylase to the point that the rate limiting step of carbohydrate absorption and digestion, just give me a second, Tiba. That the rate limiting step for the digestion is actually the absorption and not the enzyme working, because there's so much enzyme. It's very fast at digesting, but like the absorption is actually slower. This is the case for all of the everything you see in front of you except lactose, because the amount of lactase in our body is kind of little. It's quite not that much. That's why, like, if you drink a lot of milk, whoever you are, lactose intolerant or not, you'll have diarrhea. This is because our lactase is not really that good. Which part do you want me to repeat, Hiba? Okay, so dietary disaccharides. Okay, so simply dietary disaccharides. We have sucrose and we have lactose. Lactose we get from milk. Sucrose we get from, I think, fruits and like the table sugar. Simply, we said the sucrase isomaltase right here. The sucrase isomaltase, which was here and here. The sucrase part breaks down sucrose into glucose and fructose. The lactase breaks lactose into glucose and galactose, and then you can absorb all of these easily. If we have, okay, so yeah, if we have the pancreatic disease, it will affect this step right here. So we'll have amylopectin, a lot of amylopectin. We cannot like we cannot digest the amylopectin. We cannot digest the amylose. But if you have like a problem in the microvilli themselves or the brush border, you'll have like accumulation of alpha lamidextrins, maltose, sucrose, lactose. That's the idea. Depends, yani, what's affected. The step before it is basically at a red light. Okay. So that's basically everything for the digestion of carbohydrates. Are we good until now? Right. So now let's talk about the absorption of carbohydrates, and this is basically very simple. Okay, so just stay with me. When we're talking about monosaccharides, we talk about 3 monosaccharides. We have, as you can see here, glucose, fructose, galactose. Okay, and these are the ones that you will absorb: glucose, fructose, galactose. Good, good. So looking at this cell right here, we want to start from the first step, which is over here. Okay, so we have always in the basolateral membrane working sodium potassium pumps. So the sodium potassium pump will be kicking out sodium out of the cell. So we have very low concentrations of sodium in the cell. Is that good? That's great. Why? Because we use co-transporters to bring in the glucose and galactose via this sodium glucose something luminal transporter. I think that's the name. 1. All right. So the sodium glucose luminal transporter 1 right here. It is basically just bringing in sodium, and when the sodium comes in, glucose comes in with it. Basically, sodium opens the gate, and glucose just enters with it just because. All right, glucose and galactose. So this is how we absorb glucose and galactose via SGLT1, which is a co-transporter of sodium and glucose and galactose. Why does the sodium enter the cell? Because we have very low concentrations inside of the cell. Why do we have very low concentrations inside of the cell? Because the sodium potassium pump is kicking the sodium out of the cell. Cool? Great. The fructose, on the other hand, just enters through GLUT5. GLUT5 is just facultative transport. It's just a gate that fits for fructose. So this way, glucose, galactose, and fructose are inside the cell, and they exit the cell by the basolateral membrane, GLUT2. All right? I think this is very simple. The intestinal absorption of glucose and galactose and fructose. Is there any question about this? Okay. So what you need to know basically, if someone asks you what does SGLT1 do, glucose or galactose enters whatever. Fructose enters from GLUT5, and then glucose, galactose, fructose enter from GLUT2. All right, good, great, fantastic. Next up, what do we have here? So we have carbohydrate fermentation in the colon. Okay, so for the carbohydrate fermentation in colon, the idea is that the colon cannot absorb monosaccharides. Only the small intestines can. Okay, so colon cannot absorb monosaccharides. Cool. Colonic bacteria, basically, if there's anything left from the carbohydrates that reaches the colon, the bacteria will ferment it immediately. And this is mainly the cellulose, because cellulose we cannot digest it, right? So it will reach the large intestine, and the bacteria will start to ferment it. And this is why we have basically, like, this is where this bacteria gets its energy from. It's just the cellulose or whatever you left, and this is where your farts come from, basically. Total bacteria count in colon is very high, and like literally like 40% of your stool's dry weight is bacteria, which is wild to say the least. Why is there so much bacteria in your colon? Because one, the pH is nice, it's not acidic, very nice. There are no enzymes, no bile salts in the colon, no destruction for the bacteria, great. And the MMC, the motility is very slow, so they can grow much well. They can like infest the large bowels much better. Okay, and they get food basically, which is cellulose and stuff like that. Okay, so we have carbohydrate fermentation in the colon. This is just a point to keep in mind. Why is it important to keep it in mind? Because the carbohydrates in the colon, once they reach the colon, the um, bacteria will ferment them. They usually ferment them into small chain fatty acids, especially acetate, butyrate, and propionate. Propionate and butyrate both can be used to make ATP in our bodies. Okay, acetate cannot usually be used to make. Actually, yeah, it can. Acetate, butyrate, and propionate. All of these can be used to make energy in our bodies. So even though cellulose we can't really digest it, when the bacteria digest it, we get these things and we can use them to get um, basically energy in our bodies. And this is why we do not excrete this short chain fatty acids into the feces. Just less than 5% of the created short chain fatty acid will be excreted. The rest will be absorbed and used to form ATP, which is pretty nice thing for us. And the rest is, of course, the byproducts of this fermentation. We get, CO2, H2, CH3, methane, whatever. All of these will be excreted out of our body through, flatus. Let's say that. Okay. And that's basically it. Okay, so that's for this. And the last point is lactase deficiency, so lactose intolerance. What happens with lactose intolerance? It's basically the lactase enzyme is not working. Either we don't have enough of it, or there's an infection that's worsening it, or like cystic fibrosis, or whatever. There's a lot of reasons for lactose intolerance. So what's happening is that the lactose will stay in the small intestines, and and once it's reached the large intestines, the bacteria will be able to ferment the lactose much more than when you give it something else. Okay, so lactose will be fermented and basically broken down into very highly osmotic components, as you can see here: H2, CO, CH3, CO2, whatever. And all of these will act as an osmotic force that will bring the water from the blood into the large intestine. And this is why you get osmotic diarrhea. This is the basic concept of osmotic diarrhea. You have high osmolality here. It will drag the water from outside inside, and then you will basically poop water. Okay, so this is the idea of lactose intolerance. Does this make sense? We have like literally pharmacological agents that's whole work is to do this. They just cause osmotic diarrhea. We give it to people who are constipated, basically. Any questions? Each in san shi. All right. And yeah, okay. So we're still at carbohydrates. I don't know why they talk about probably because it's important. Okay. So carbohydrates. Last thing is this test. So this test we do in case we want to test for anything that is like there's a problem in the digestion and degradation of disaccharides, okay, or any sugar, basically. What we do is we give the patient the thing we suspect that they cannot digest. So in case of lactose intolerance, we give them lactose. After a few hours, we measure the hydrogen that's coming out of their mouth and their breath. Okay, the idea is that if the lactose stays until it reaches the large intestines, that means it's not degraded. That means we have lactase deficiency. The bacteria in the large intestines will ferment it. and fermentation will produce a ton of hydrogen gas, and this hydrogen gas will leave through your mouth. So if you have high amounts of hydrogen gas in your breath, this means that your body has a problem digesting this lactose. Okay, we can also do blood tests. Yeah, and we can do this basically for anything. So if we suspect that someone has a problems in, I don't know, maltose degradation, we can give them maltose and then check their H2. If we suspect sucrose problems, we can do the same thing, and etc. So it's not specific for lactose. The only thing that's specific for lactose is that you give them lactose and then you measure H2. All right, does this make sense? Of course, we can measure, as you can see here, we can then check the blood test, then we give them lactose, and we check the glucose levels. We would expect that the glucose levels should rise if they have normal lactose absorption, you know? But if they don't have normal lactose absorption, the glucose will not rise. I think this is the last point about the carbohydrates, which is the congenital sucrase isomaltase deficiency. So we said this is one enzyme that does two jobs. Sucrase will break sucrose into glucose and fructose. Isomaltase will break the limit dextrin of the amylopectin, right? The alpha 1,6 in every limit dextrin. So a deficiency of sucrase isomaltase enzyme will mean that we cannot break down sucrose and we cannot break down amylopectin, or like specifically the limit dextrins of amylopectin. Okay, so we will find a lot of limit dextrins in the stool. That you may be able to find that actually. Yeah, and basically in the pediatric population, because most of these things you will notice in the pediatric population, in the babies basically, once you start basically giving them juices that contain sucrose, you will catch this disease. Because before that, they're only drinking milk, which has lactose. But like once you introduce sucrose, you will see this problem. And it will present the exact same way as lactose intolerance because it's basically the same thing. It's just osmotic diarrhea. And how you test it, you do the same exact test, but you give sucrose rather than giving lactose. And here he's just saying, oh, this same concept, we use it to induce, basically, to treat constipation through dietary fibers, cellulose, or like digestive biscuits. And that's it for carbohydrates. Are we good until now? Okay. Inshallah, I don't take more than half an hour with the rest, but if I do take more than half an hour, sue me, please. All right. Hydrolysis of ingested nutrients. We have proteins. So proteins are degenerated or degraded in the lumen, the microvilli, and the enterocytes. Cool. So it's done in every single area. We have pancreatic proteases, cross-border peptidases, and cytoplasmic peptidases. Cool, great. Now, one thing you need to like keep in mind is that not all of these proteins that we will be degrading and absorbing are coming from your food. In fact, almost 50% of the proteins that you will be absorbing and degrading are proteins that you are secreting yourself. How? Well, all of the enzymes that you are secreting, all the pancreatic enzymes, all the gastric enzymes, all of these, they will need to be they will need to be degraded and absorbed just like they are food, because basically your gut doesn't really differentiate between an enzyme that you secreted and some proteins you got from like a cow. It's the same thing for it, so it will be just degrading it all and absorbing it all like its nutrients. So all the enzymes, the mucoproteins, the mucus, all of these, and even the epithelial cells that are dying and shedding, you will break them down and you get a ton of good proteins that the cell will want to reabsorb, So yeah. So the sources are both from the diet and the endogenous proteins. Great. Okay, now the thing about protein digestion in the lumen. So in the stomach, we're talking about the stomach here. we do about like 10 to 15% of all of the digestion, which is not a lot. 10 to 15% is not a lot. And even like, as you can see here, people who had a gastrectomy, so they removed their entire stomach, or like they cannot secrete acids, which basically screws over all the protein digestion in the stomach. These people literally have no noticeable problems in protein digestion. They have no noticeable problems in protein like levels in the blood. That means that the main place for protein digestion and absorption is the small intestines. Okay? And where do we get the enzymes from? From the pancreas, of course. Okay? Okay, so unlike what we had in the carbohydrates, proteins can be absorbed as like peptides, like dipeptides, tripeptides, and amino acids. Okay, so we don't really have to break them down into like teeny tiny amino acids. What are the enzymes we're talking about when we talk about like pancreas juice? We talk about mainly trypsin, chymotrypsin, carboxypeptidases, and elastases. Okay, now um, all of these are secreted. I think you took it in one of the lectures. All of these are secreted in an inactive form, and of course, they have to be activated by either the trypsin that has been already activated or the enteropeptidases that are on the brush border. Yeah. Okay. So another thing to note is that trypsin and chemotrypsin are endopeptidases, so they cannot break down the terminal bonds. Which, what does that mean to us? That trypsin and chymotrypsin will only leave you with di and tripeptides. They will never be able to leave you with an amino acid. Okay? Peptides are longer than di and tripeptides. So di, tri, and then like longer peptides and amino acids. So trypsin and chymotrypsin and pepsin, all of these cannot leave you with just pure amino acids. To get pure amino acids, you need carboxypeptidases A and B. Carboxypeptidase basically means that it cuts the peptide at the carboxy the C terminal, the carboxyl group. In our bodies, we do not have like an aminopeptidase, an amine group. No, we only have the carboxypeptidase, and elastases are also intropeptidases. Okay, so mainly we will get di and tripeptides. We will get some amino acids, and we'll get basically very little peptides. That's what happens in the lumen by trypsin, chymotrypsin, carboxypeptidase. Is this clear until now? Okay. This point again, just the main thing that you need to know about this point is that in the lumen, what is breaking down the proteins? Trypsin, chymotrypsin, and carboxypeptidases A and B. These are coming from the pancreas. Stuff in the stomach, not that important, but again, so the trypsin, chymotrypsin, and pepsin, all of these are endopeptidases, so they cannot leave you with pure amino acids. We don't have any pictures of amino acids here, but yani, if you imagine, it's the same thing as we had over here, right? So if you imagine these are amino acids and these are endopeptidases, so they can only break down here, they cannot break down here. So they will always leave you with like 2, 3, and not 1 alone. Okay? And that's what an endopeptidase is. A carboxypeptidase will leave you with just 1 amino acid because it's an exopeptidase. And they break the peptide at the carboxyl end. That's basically what we're saying here. And what will be left, and after all the gastric and pancreatic juices are done, a lot of di and tripeptides, because trypsin and chymotrypsin are very much more than carboxypeptidases. Some amino acids, because again, carboxypeptidases are working, and very little peptides, because like you should be done by from the peptides by now. Okay, so that's the luminal protein digestion. Now we have the brush border and the intercellular. They're very simple here. We have brush border peptidases, which break down the peptides into di and tri and even amino acids. So right here, this peptidases, different types of peptidases, they take all of the oligopeptides and then just cut them down. They cut them down into amino acids and di and tripeptides. Cool, great. We have transport systems. They are kind of different between di and tripeptides right here. This one basically lets di and tripeptides enter. This one lets amino acids enter. So we have transport systems, and we have cytoplasmic peptidases. So even the di and tri, once they enter the cytoplasm, they will still be cut into amino acids, because you cannot like go into the blood unless you are just pure amino acids. You cannot like di and tripeptides cannot enter blood. They need to be amino acids. Okay. See, the brush, unlike the pancreas, all right, the pancreas does not have only has carboxy peptidases. The brush border has amino and carboxy peptidases, so it is much more proficient and much more equipped to give you amino acids and not just di and tripeptides. Is this clear? Yes, sir. Grant. Okay. Now just a bit of detailing about how the absorption happens. As you can see here, this is the can you repeat? I'll repeat after this point. Just tell me what you need exactly to repeat. So amino acid transport, as you can see here, again we have the sodium potassium pump kicking out the sodium as much as possible all the time. It's being kicked out. Why? Because we want very little sodium in the cell. Why? Because the amino acid transport works via a co-transporter with sodium. So it requires sodium amino acids to enter inside. There are different carriers in like different transporter, different carriers. Some are good for neutral amino acids, some for acidic, some for basic, whatever. You have enough for everything. Okay? And just the idea is we keep the sodium level low here so that sodium will enter and amino acids will enter with it. Okay? Now, defects in the amino acid transporters are very, very, very rare. One of them, but this is more important for renal causes, is cystinuria, which will cause you kidney stones. And it will be, and it will also have a bit of problems in the intestine, but like not that big of a deal in the intestine. But in the kidney, it causes kidney stones, and you will study it more in mole, I think, and maybe in renal. I'm not sure. And that's for amino acid transport. Very easy. Sodium dependent mechanism. The mechanism for the peptides is not sodium dependent. It's actually hydrogen dependent. So what we have here is, again, we start from the beginning, which is we have a sodium potassium pump kicking out sodium. Great. So we have low sodium here. But this time we want the low sodium here because we want to use the sodium hydrogen like exchanger. So we get sodium inside and we push hydrogen outside. Now we have high hydrogen here, low here. So the hydrogen wants to go back in, and it goes back in through the peptide transporters. These peptide transporters will allow, like when hydrogen enters, di and tripeptides will enter as well. And then the di and tripeptides will be broken down into amino acids via cytoplasmic peptidases, and they will exit into the bloodstream via different, right here, different transporters. Okay, I think this is like not very difficult to understand. This is just basic stuff, right? Inshallah, is it clear? Good. Okay, Maya, do you have, what exactly do you want me to repeat here? Okay. All right. Great. Alhamdulillah. So until now, if we want to review like just protein digestion real quick, just review it real quick again. We have pepsin, trypsin, chymotrypsin. Our main ones are endopeptidases, so they will leave us with dry and tripeptides. We have carboxypeptidases A and B from the pancreas, which will leave us with amino acids, hopefully. And this is our gastric and pancreatic, our luminal hydrolysis. Cool, great. It will leave us with mostly di and tri. Brush border has peptidases that will break the peptides into di and tri and even amino acids. Di and tri afterwards will enter through the hydrogen dependent mechanism, which is being fueled by the sodium hydrogen exchanger. The amino acids will enter the cell via a sodium dependent mechanism. The di and tripeptides will be broken down into amino acids. Amino acids will leave the cell into the blood. Okay. And that's basically everything you need to know about the protein assimilation of proteins. Now, one thing, one application of all of this is that these peptide transporters allow anything that kind of looks like a peptide to enter. Okay, so a lot of drugs that we use actually kind of sort of looks like peptides. So a lot of antibiotics like penicillin, uh, a lot of antibiotics, a lot of antihypertensives like angiotensin converting enzyme inhibitors, ACE inhibitors. Um, like we have a lot of a lot of drugs that that look like peptides, so they enter through this transporter, and then they just go through their drug transporters into the blood. And even some of these drugs actually are prodrugs, so they require to be activated, and they are activated by different enzymes in the enterocyte. This is just, as you can see, it's of relevance in pharmacotherapy, and I think you will take this in POD, like in the first or second lecture of pharmacology. So we're done with proteins now. What time is it? All right, we're done with proteins. We're done with carbohydrates. Now with lipids. Okay, so lipids, they need both pancreatic enzymes, as you can see here, but also they need bile. All right, and this is the end. This special thing about them. Okay. All right. So where do we get our lipids? We get them mostly from the diet. They are mostly long chain fatty acids. Some of them are medium chain, but usually you will get only long chain fatty acids from your food. Okay. Now we have a bit of gastric lipases, as you can see here, but they're not that important for adults. They are important for babies. Babies need gastric lipases to be able to, digest lipids well. And if an adult has a pancreatic insufficiency, again, they require gastric lipases. The gastric lipases are very, very helpful. Otherwise, they're not that nice. Okay, they're not that important. But the thing about them, and they do not require, as you can see, they do not require a colipase, nor do they require bile salts. So gastric lipases do not require bile salts, do not require colipase. Cool, great, fantastic. Now let's talk about pancreatic lipases. Pancreatic lipases they target all triglycerides, and they require bile salts and colipase. Okay? Now, the bile salts come from where? They come from the liver. I think you took a lecture about the liver, like physiology of the liver. They basically have two functions, which is emulsification and solubilization of the TG, the triglycerides, and the long chain fatty acids, respectively. Okay. Now we will talk about it in more detail. So we have here the process of emulsification. Now, what is the process of emulsification exactly? So keep the idea in your mind that the the lipase that's coming from the pancreas, okay, it can only work on the surface, right? So this is a fat globule. It can only work on the surface, okay? So if I have like this large fat globule and I can only work on the surface of it, all of this inside, the lipase cannot work on it until it's done with these over here, and then it will go down and down and down. That takes so much time. All right, so that's very, very inconvenient. So what we do is we use these bile acids. The bile acids have a hydrophobic side and a hydrophilic side. So the hydrophobic side will basically get attracted to the lipid because the lipid is also hydrophobic, and the hydrophilic side will face the lumen because the lumen is full of water. Great, fantastic. So what happens is that it basically does the emulsification, which is it just breaks down this fat globule into smaller and smaller and smaller little droplets, emulsified droplets. These droplets, as you can see here, they're small, and that means that there's not much inside. The surface area is now much larger. If we break down this 1000 micrometers diameter into like multiple ones with 50 micrometer, the surface area will enlarge much more, and this means that the lipases can work much better because you have more surface area. I think this makes sense, right? So this is the entire like logic behind emulsification. Why does the body emulsify? Because it just wants to increase the surface area of the fat so that the lipase can interact with the fat much better. Simple? Easy peasy. Yeah, okay. So that is emulsification. We're done basically with emulsification, and it's done by bile salts and lecithin. Lecithin is a phospholipid. Okay? Okay. Now the next step. So yeah, this is what I was just saying right now. It's lipases can go to the interface and cut it down, and of course, the pancreatic lipases require colipase. What does colipase do? Colipase basically just grabs the lipase and basically allows it to attach on the droplet on the surface. So without the presence of colipase, the lipase cannot anchor here and cannot work. That's basically it. So pancreatic lipase requires the colipase, and colipase is activated by trypsin. All right, so now that is our emulsification. Now for the production of solubilization via micelles. So now we basically broke down these triglycerides. We broke them down. Once you break down triglycerides, as you can see here, you break it down into free fatty acids and one monoglyceride. So two FAs and one MG. Makes sense, I think. So these fatty acids and this monoglyceride basically will be inserted into a micelle. A micelle is basically very similar to it just has these bile salts all around. Again, hydrophilic outside, hydrophobic inside, and they are much more smaller than what we were talking about here. So they can actually melt. They can actually be solubilized inside of the luminal fluids. So they will solubilize and they can be absorbed because the fluid is absorbed. So anything solubilized inside of it will be absorbed. Like if it's salt, it will be absorbed. So the micelle will also be absorbed. And that's the way basically you get the free fatty acids and the monoglycerides inside of the enterocytes through absorption, through solubilization into micelles, or like using micelles to solubilize the fatty acids and monoglycerides in the water or like the fluids and then absorb. Is this simple? I think you guys already took all of this. So micelles. So again, micelles are just simply the same concept here. Here, why did we do this? We did this to increase surface area. Here, we just made it much more smaller so that it can be solubilized. I think that's a word. Solubilized. Melted. They can melt in the water. Okay? Just like your salt, it is a word. Thank you, Tariq. They can be solubilized in the water. So the micelle that contains the fatty acids and the monoglycerides that have been broken down from the triglycerides now can just be solubilized into the water, and then you absorb the water normally. It's just what's inside of your bowels, so it's just absorbed easily. That's the whole idea. So you make it small so it can be solubilized, because this will only be suspended. This can be a solution. Make sense? Right. So that's our micelles and everything. Okay. Here, basically, it's just telling you that. So one thing just to note is that all of this we're talking about is when we're talking about the long chain fatty acids. Now, medium chain fatty acids, they can go through this process, but also they can just diffuse into the cell, no problem. Like they can just enter the cell, no problem. And this is why if you have someone with pancreatic insufficiency, they do not have a working pancreas, their diet is usually medium chain fatty acids, because the medium chain fatty acids does not require breaking down. They immediately just enter into the cell, no problem. Okay, is clear? It's not something you will need to know, but I think it's in the notes section uh, of the slides. So just the main note that you need to get is that the long chain fatty acids are not soluble in the chyme itself. So they need to be micelles in order to become solubilized, and then they will be inserted in, or like incorporated inside of the epithelial cell. If there is no bile salts or there is problems in fat absorption, you will be pooping out fats, which is steatorrhea, which is basically you'll find your feces to be very yellow, whitish, and it smells awful. And that's all I know about it, and hopefully I don't need to smell it in my life. So yeah. So that's for this here. Now, in general, all fatty acids in our bodies are like stuff that we eat will be esterized. So, or fats. So, like right here, we have cholesterol. The cholesterol that we eat will be a cholesterol ester. So it has a fatty acid like bound to it. So through this enzyme cholesterol esterase, we break it down and we have a cholesterol alone and a fatty acid alone. Okay. And then here you have a phospholipid, which is lecithin. Again, it has a free fatty acid, fatty acid, and this thingy. What was the name? It's written in this in the note section. Anyways, so we have this, and through phospholipase A2, we break it down into a free fatty acid and this lysolecithin. So the whole idea is that everything lipidy that we eat is esterized into fatty acid, and we break down this fatty acid to get a free fatty acid, and the whatever is remaining can be incorporated usually into a micelle and then absorbed. Okay. Now, as you can see here, this was all just the digestion and the breaking down of the lipids. We talked a bit about the absorption here, but again, absorption, the main point about absorption over here. We over here have our cholesterols, monoglycerides, our whatever this is, lysolecithin or lysophospholipase or phospholipid, and free fatty acids. All of these will be incorporated into our micelles, our very small, tiny, cute micelles. All right. These can just immediately enter into the cell, no problem. We don't need any transporters. We don't need anything. We just, because it's fat, and the the cell membrane is pretty nice to fats, so it will just let the fat enter, no problem. And now we have our cholesterol, monoglycerides, our lysopl, whatever, and our free fatty acids inside of the enterocyte. Now, one thing that will happen is that we will recreate what we broke down. So our cholesterol, we will add to it a free fatty acid, we get our cholesterol ester. Our monoglyceride, we will add to it 2 fatty acids, and we will get our triglycerides. Our lysophospholipid, we will add another fatty acid, and we will get our phospholipid. So we will make what we broke once again. And this happens, where does it happen? It happens in the smooth endoplasmic reticulum. Okay, so the smooth endoplasmic reticulum will be forming or reforming the broken down fats. Why? Because we want to incorporate them as they are into the chylomicron. Now, what is the chylomicron? The chylomicron is just the way that the fat will be transported from the intestine into the liver. Okay, so again, they're fats. They cannot be just swimming around willy-nilly. No, they will be very damaging to you if they are just in your bloodstream or just in your lymphatics. You cannot have free fats, so you need them to be covered in something, and that's why we use the chylomicron. So the phospholipids will act as the the borders, or like the membrane of the chylomicron, and the chylomicron will need to have this apolipoprotein B48. I think you will take it in mol 2. You will take like a full lecture about what the hell is a chylomicron and what the hell are lipoproteins in mol 2, I believe. But yani, the idea of ApoB is it's just basically the passport for the for the chylomicron. As long as the chylomicron has ApoB48, your body knows that, oh, this is a chylomicron. Without it, there's no ID. It's not it's ID basically, not a passport. It's ID. And then you have inside of the chylomicron, you have the cholesterol esters and the triglycerides. All of these will exit the cell and enter the lymphatics, and through the lymphatics, they will reach the liver, They will enter the liver, and then the liver will do the sorting out, and basically it will package it in another package, and will send it out to the body like the body needs, because the liver knows what's up. Okay, so this is everything for the long chain fatty acid. For the short and medium chain, they just go from the lumen into the blood, no problems. And this is again why, if we have like problems in these enzymes and these bile salts or whatever, we just give them medium chain fatty acids and we're good. Okay, is this clear? Fantastic. All right, now for the miscellaneous stuff. So we have lipid soluble vitamins. How do we absorb lipid soluble vitamins? We just put them here. So we have cholesterol, Mg, whatever. Over here, you'll find like a vitamin A, a vitamin D, a vitamin K, whatever. They can enter, no problem. They will enter, and then they will be packaged into chylomicron and kicked out of the cell, no problem. So lipid soluble vitamins are basically treated just like lipids. Nice, nice. What about water soluble vitamins? Water soluble vitamins need their carrier mediated absorption. So they need their specific carriers, and usually they are sodium co-transporters. So for example, vitamin C. You already took the vitamin lecture, right? From Mall. I think this week it is. Yeah, you took it. So you know which is lipid soluble and what is not. What is water soluble? The lipid soluble are the the DEAK, and the water soluble are the B and C. So C, as you can see here, it's just telling you, no, C enters into the enterocyte via this whatever transporter. This is its unique transporter, sodium vitamin C co-transporter, and then it is kicked into the blood, no problem. So that's it for vitamin C. Okay, so that's our vitamin C. So lipid soluble vitamins are treated just like lipids. Water soluble vitamins have specifications. Each one needs its own carriers. Vitamin C has the SVCT1. Vitamin B12, this one is very important to know. And honestly, I remember like getting a lot of questions. Like wherever this comes up, it's important, because vitamin B12 is quite important when we talk about like the absorption. It's just because it's such a unique mechanism, so they always love to ask about it, because wow. So what is happening here? Vitamin B12, you get it from food, and you get it from food. Usually it is bound to different proteins. Okay, so when you eat your food, you get it, as you can see here, it's bound to different proteins. You have a secretion from your salivary gland called a haptocorin. Okay, so the haptocorin comes from the salivary glands, from specific whatever the salivary glands. Now, this vitamin B12 will go down into the stomach. In the stomach, your pepsin will be working, your acids will be working to break down this protein. So the protein that's complex to the B12 is now broken down, and now you have the B12 alone. B12 cannot survive by itself, right? Especially in the intestines. It is immediately destroyed in the intestines, okay? So it needs something to protect it. What protects it in the stomach itself? It's the haptocorin. So the haptocorin comes in and binds to the vitamin B12, and now the vitamin B12 can, in the haptocorin complex, will go into the intestines. Now, in the intestines, the haptocorin will be destroyed. And now, again, the vitamin B12 is in danger of getting destroyed. So what protects us this time? The intrinsic factor. And this is really the thing that you really, really, really need to keep your mind on. The intrinsic factor. Haptocorin is like a new thing. It was literally discovered like I think a decade ago or something like that. But the intrinsic factor is something that you really need to keep your mind on. Why? Intrinsic factor is secreted by what? By the parietal cells of the stomach. So the cells that secrete the acid also secrete your intrinsic factor. This intrinsic factor simply just goes into the intestine, and again, it binds to the B12 and it protects it from getting degraded. That's one. Two, it allows it to interact with the receptor that acts as its transporter into the cell. So without the intrinsic factor, B12 cannot interact with its transporter, so it cannot be absorbed, so it will be excreted. Does this make sense? I think we're okay. Okay, so just the simple idea is that our intrinsic factor is very important for us to be able to absorb vitamin B12. Okay, and where does the intrinsic factor come from? From the stomach. So literally, I think you took like the gastric secretions in like the second lecture, and surprisingly enough, out of every single gastric secretion, okay, the most crucial one and the one that cannot be replaced at all by anything the body does is intrinsic factor. So anyone who has a total gastrectomy, so you remove the stomach, they digest food fine. They can protect against bacteria. Yeah, not as well, but they're still protecting against bacteria pretty well, no problem. The only thing that they cannot do, khalas, is absorb B12. They just cannot absorb B12, no matter what. Anyone who had a gastrectomy, uh, because I don't think we have like intrinsic factor, like B12 intrinsic factor, like I don't know, why can't intrinsic factor be replaced? Why would you want to? The question is, why would you want to replace intrinsic factor? Because like, if I remove the stomach, yeah. So now I'm going to make my dietary I cannot eat anything I eat. B12, I cannot eat it. Right? That's number one. I cannot absorb B12 that I have eaten. And if I want to make a drug that has both vitamin B12 and like an intrinsic factor together, and then I give the patient, okay, this is your drug now, and this you will be able to absorb this. I mean, why would I do it when I can just give them like B12 injections? There's no need for the extra step of making intrinsic factor, like making a pharmacologically available intrinsic factor. I'm not sure if it's available or not, but the idea is no, the most essential secretion from your stomach is intrinsic factor. That's the idea that I want to give you. It being replaced or not replaced, it's that's like beside the point here. Okay, so that's intrinsic factor, and then they basically are dissociated from each other in the in the lysosome, I believe, and the intrinsic factor, the vitamin B12 is transported through the blood via transcobulamine into the liver, and basically stuff happen in the liver as always. So that's our vitamin B12. I hope it's clear. Vitamin B12 is very important in like anything. In MOL, you will be asked about it. I think in GIT, you will be asked about it in first year and third year. It's very important. For folate, on the other hand, folate is much more simpler. You get folate in your diet as this thing, PTER with 7 glutamates. So 7 glutamates are like just onto it. And through this folate hydrolase on the brush border of the proximal small bowel. So the proximal small bowels are the ones that will be degrading and absorbing, I think, folate. Only the proximal. While this, just one thing I really need to note here is that the absorption of B12 can only happen in the terminal ileum. Only in the terminal ileum. If you remove the terminal ileum of a patient, they cannot absorb B12. If you remove their stomach, they cannot absorb B12. So stomach bad, terminal ileum bad. Okay? And any problems in the terminal ileum, they cannot absorb B12. Here, this is the proximal small bowels, and this is where folate basically is done. So through this enzyme, folate from polyglutamate to monoglutamate, and then it is absorbed via a hydrogen-dependent mechanism, this PCFT right here. And again, the hydrogen mechanism, what feeds it? Hydrogen sodium exchanger, just like your typical protein or whatever. Okay, so the proton coupled folate transporter fueled by the sodium hydrogen exchanger. That's for folate, and this happens in the proximal small bowels. Mostly, I think it was jejunum. I think it's jejunum. Okay, now the last point we have over here is these two. What happens if we cannot absorb them? We get deficiencies. And what we get is a very specific type of anemia called a megaloblastic anemia. The megaloblastic anemia basically have less red blood cells, but each one of them is very large. Okay, so you have a large red blood cell, but they are very few in numbers. And why does that happen? It's because both B12 and and folate are both important in DNA synthesis. So when you have like less amounts of them, you will have worse DNA synthesis. So it will take you longer time to synthesize the DNA, and that's why the cell grows larger than it is intended to be. You don't need to know this. These stuff, this especially will be taught much more in the heme block, HLS. But just the idea, there is this type of anemia called the pernicious anemia. Okay, the pernicious anemia is caused by an autoimmune disorder that destroys, what does it destroy? The parietal cells. The parietal cells are destroyed. Intrinsic factor is not produced anymore. B12 is not absorbed anymore, and that results in an anemia that is megaloblastic. Okay, and how do we treat it? We give them vitamin B12 intramuscular injections. Okay, I think this is a mistake. I think oral vitamin B12 is ineffective because right here it says about 1% absorbed in the absence of intrinsic factor. So I think this is a mistake on the doctor's part. I'm not sure. I can't be very sure about it, but I think it's a mistake. On the other hand, we have folate deficiency, which again is the same thing. If you have a folate deficiency, you'll get a megaloblastic anemia. And how do you treat it? You give them folate supplements. Folate deficiency is very bad for females who want to be pregnant because it is a major cause of neural tube defects. So they might be born without a brain, or with a messed up brain, or a messed up spinal cord, or a messed up spine. So I think everyone knows folate supplements before pregnancy. It's like the first thing you know about anything medicine. And yeah, so that's vitamin B12 deficiency, vitamin B9 deficiency, folate deficiency. And I think, and those are just review. I think that's it for this lecture. This is here, he's just telling you why, like, breaking down of lactate will make it worse, because lactate alone is like 1 osmotic power, and once you break it down, it becomes 6 osmotic power, so it becomes, osmotic diarrhea. And these are the main symptoms of osmotic diarrhea, bloating, pain, gas. And these two are very nice review slides. This is for the fat, and this is for the amino acids. And yeah, I think that's all. Does anybody have any questions? Great, alhamdulillah. I think everyone has my number, but like if you don't have my number, I'll just write it down. Yeah, right here. So this is my number. If you have any questions, feel free to ask me any questions. Among like everything in first year, I remember CVP, cardiovascular well. I remember GIT very well. I'm really good at MOL, alhamdulillah. The rest, give and take. I can like read up. I send it in the chat. You can open the chat, Yeah. All right, let me stop sharing it. Yeah. If you want to ask me like any questions, I'm mostly free. Alhamdulillah, my block right now is kind of a chill one, but I do need to focus on studying more. Yeah. If you have any questions, feel free to ask. And